Good evening, everyone. This work is jointly done with Shaptoshi Dasgupta, and I'm a postdoc with Professor Madan Rao at NCBS. The topic is active force patterning in a mixture of contractile stresslets. So I'll first explain this word, stresslets. Yes, so what are these stresslets? We know that inside cells we have various mechanotransducers like various motor filament complexes. Examples include these contractile actin myosin complexes, extensile kinesin microtubule complexes. So these motor filament complexes, they both act as force generators through activity and also as force sensors. So this term is missing, sorry. So force sensors. So the force sensing ability comes from their presence as either catch or slip bonds through strain dependent binding unbinding onto the meshwork. This dual activity of these mechanotransducers, so we are calling these elements as stresslets. So it is known that these molecular stresslets, they have different cellular localizations. So they are present, and so they are present at various levels at different parts of the cell, and their activity levels and their binding unbinding rates, they are also different. So due to this localization of molecular stresslets, what we see at the macro scale of the cell are these stress fibers. So these are like patterns of stresses which spans the entire scale of the cell. So this study is about how this mapping happens. So the localization of molecular stresslets at the micro scale. From this localization, how do we get this patterning of stress at the macro scale of the cell? So this study is about this this mapping. So to get this patterning of stresses at the macro scale, the first requirement is we should have a spatiotemporal segregation of a uniform mixture of these molecular stresslets. So to study this spatiotemporal segregation, so we take a simple model consisting of just contractile stresslets such as myosin 1 and myosin 2, and these two stresslets are undergoing binding unbinding on the actin meshwork. So these are the densities of these elements, and it has also this unbound species and of course a background cytosol which is fluidic. So in the time scale we are interested in, we are just focusing on the dynamics of this meshwork and this bound myosin species which are getting undergoing binding unbinding onto this actin meshwork. And in our time scales, so this actin meshwork we are considering is as an elastic network. So the first so this is a hydrodynamic model. So the first equations that we write down are for this actin meshwork which is elastic. So the first equation you are seeing here, this is an overdamped dynamics where u dot is the velocity of this meshwork and sigma is the stress field, and this the density of the actin meshwork. So we are assuming that it is long lived. So there is no diffusion and turnover term present here. And so this stress field you are seeing here, so this is an active gel model. So it has this primarily two parts, passive and active. So the passive parts has elastic and viscous contributions, and the elastic part comes from a free energy density, which is like this is a quadratic energy density. So it means that the elasticity is linear elasticity, and the active part depends on the density of the individual this myosin species. So here for simplicity we are taking it as a linear function, and it can also implicitly depends on the meshwork density rho a, so through this function chi. And of course there is a viscous part which is passive. Now so this completes the dynamics of the meshwork. Now next is the dynamics of the stresslets, these bound myosin species. So this is just a reaction diffusion advection system. So these myosin species, they gets advected by the velocity of the meshwork. And so just for simplicity, they have the same two diffusion coefficients. And this term is the binding unbinding term. So this binding unbinding term, so since these bound myosin species, they act also as a stress sensor. So this unbinding term depends on the strain. So here we are taking a exponential form for this unbinding rates, and these numbers you see here, alpha 1 and alpha 2, they control whether the bond is a catch bond or a slip bond. So catch bond means like if you pull the meshwork, the species will unbind, and the slip bond is just the opposite. So so we have this 1, 2, 3 equations. We combine these equations into a system of nonlinear reaction diffusion advection system, and here we are presenting only the scalar non-dimensionalist form for simplicity. And we have also recast the equation in terms of these two parameters, rho and phi. So the average density and the relative density of the stresslets. And this relative density is our segregation order parameter. So we are going to study segregation of this system. So phi equal to 0 means it is a uniform mixture, and that uniform mixture should be unstable to have segregation, spatiotemporal segregation. So the stress field you see here. So when you do the complete algebra, so remember we started with a linear elastic solid, but due to presence of activity, so the effective elasticity is becoming nonlinear. And these terms you see here, so there is a backstress term which is purely active, and the linear elastic moduli is also getting renormalized by activity, and two new moduli are appearing which are purely active in origin. So one can explain the form of this stress field through an effective strain energy density landscape. So remember that this is an active solid, so there is no free energy for the whole system, but this is just the elastic part. So so these two moduli B2 and B3, they basically what they are doing, they are putting these two walls. So one wall for no self penetration, so it cannot like shrink infinitely, and another wall is no crack condition. So this is the model. Now we will study the linear stability of the homogeneous unstrained uniform steady state of the system. So which means we are starting with an uniform mixture. And so we did we perturbed the system about this steady state, and we get a dynamical equation with this dynamical matrix. And as we know that the general solution for distinct eigenvalues has this form. But if you notice carefully, so this is an active system. So this dynamical matrix is non-Hermitian basically. So non-Hermitian dynamics means we have non-orthogonal eigenvectors. And so there will be cases when some of the eigenvectors may collapse onto each other and eigenvalues also coincide. So this brings in an interesting dynamics, interesting picture in this whole story, because in usual Hermitian dynamics we are restricted to only normal eigenspace. So next, from the eigenvalue analysis of these eigenvalues, we get the phase diagrams, and as you see that so this YOLO regime is the segregation. So there is a very prominent segregation regime, and so segregation happens when the stronger stresslets, they unbind slower. So they are trying, the stronger guys are trying to stick to their location. So starting from a uniform mixture, so you get like segregated phases. So phi positive, phi negative sites. And another interesting phase that we get is traveling waves. So this happens at the boundary between the growing wave and damped wave phase. So for traveling wave, the stronger guys must unbind faster. So the traveling wave phase, it breaks both the time translation and the space translation symmetry. Whereas there is another phase here called which we call the swap phase. So in the swap phase, this is also an oscillation, but this is not traveling. So this is a standing wave. Yeah. So the swap phase occurs when the stronger stresslets unbind before the contractile instability kicks in. So yeah. So we asked what is the driving force behind this segregation. So here we are finding out the elastic stress distribution is behind the segregation dynamics. So but it is only in the linear regime. So this is the effective strain energy density in the linearized for the linear regime. And so you see that as segregation is progressing, so in this regime, so the stress power is dropping sharply. So this is the minimum elastic energy dissipation basically, which is driving the segregation. Now we ask what is the late time state of the segregated domains. So we get this segregation, phase segregation. So what happens to the segregated domain? So in usual phase segregation dynamics, it is a coarsening dynamic. So these domains grow and it forms a bicontinuous system. But here, so across this line, so this is the profile. So this green line is the stress profile, and from the equations, you know that the stress jump basically causes the velocity. So what happens here is this front and this front, they move towards each other due to their opposite stress gradients, and as a result, instead of bicontinuous configuration, you get singular lines of the stronger guys. So the stronger guys eventually form singular lines on the domain, and these singular lines are akin to the stress fibers that we see. And of course, in reality, there is no singularity, but it is always suppressed by this steric hindrance put here through these walls. So just to conclude, so we get an active version of the Young-Laplace law, like what is the curvature of this line and how it is related to the pressure difference across the lines. And I am going to skip this. So this is about the non-normal dynamics I was speaking about. So I'll just skip. So just to summarize, so activity and strain dependent turnover leads to spatiotemporal segregation, and the stress dissipation is driving the segregation, and nonlinear feedback. So it leads to stress singularities basically, and I'm not going to discuss this. Thank you.
We have time for 2 questions.
I have 2 quick questions. So the first thing is, in your model, you have only actin fibers and myosin motors.
Yes.
So you have other, in a normal cell system, you have other actin polymerization, depolymerization adapter molecules, which can actually have an impact on what you are modeling. So have you considered considering
we are in the process of doing this. So this is just a simple model with two elements. Okay. So 2 elements are separating. So if we increase the elements, so it becomes very complicated. And also the actins, they are not really, so the active stress here is isotropic.
Okay.
So in reality, it is actually polar. So we are considering all these facts.
So this model that you have, even if it is a simple model, have you ever tried to extend it to experimental model where you can artificially create biomembranes or something to check whether the stress, whatever you are calculating the Young's model is, everything, whether it's actually experimentally producible? Have you ever tried?
Not yet. So stress fibers has been observed for a long time. So we are not correlating directly with our model to stress fibers. So stress fibers are dynamic, and so it also remodels.
Stress fibers you can't correlate because you have much more variables. But the model that you are actually considering, you can actually recreate that in a lab, right?
Yeah.
Because of the limited parameters that you are considering. But even that, have you ever tried?
No. So we have been thinking of doing experiments. So let's see. Yeah. Thank you.